Hello everyone and welcome. This is variant 2 of May-June 2024 9700. Before we get started, make sure to check out the free notes I have available for you in the comments down below. There will be more notes released soon, so make sure to subscribe to stay updated. Now let's get started. Question number 1. A prokaryotic cell, which is 1 micrometer in diameter, is magnified 50,000 times in an electron micrograph. What is the diameter of the cell in the electron micrograph? Okay. He's asking for the diameter of the cell. He gave us the actual size of the cell and he's looking for the image length. Okay, let's remember the triangle. The triangle says we have magnification, image length on top, and actual length right next to it. So if we want image length, then we should multiply magnification by actual length. So in this case, we should multiply 1 by 50,000, and this gives us 50,000 micrometers. Now he's asking for the answer in millimeters, so you have to divide by 1,000 to convert from micrometers to millimeters. You get 50 millimeters. In other words, 5 times 10 millimeters. The diagram shows a plant cell with some labeled structures. You have P, S, R, and Q. Which structures are bound by double membrane? Okay, double membrane, we're going to have the nucleus has a double membrane. The chloroplast has a double membrane. The cell wall does not have a double membrane. Neither does the vacuole. So obviously we can here pick P and Q. Which size of ribosome is found in mitochondria and the typical prokaryotic cell? They both have 70S ribosomes, which are 80 nanometers in diameter. Which row about typical prokaryotic cells and typical animal cells is correct? Okay, I have lysosomes present for the breakdown of old organelles, and ATP is produced by the cell. Actually, prokaryotic cells do not have lysosomes, because they don't have any membrane-bound organelles, and so we can immediately cancel out the first option. So here we choose an X. ATP is produced by both prokaryotic cells and animal cells. Despite prokaryotic cells not having mitochondria, they do have these inner foldings of their membrane called mesosomes, which contain enzymes, respiratory enzymes, which are able to produce ATP. So C. Which row is correct for the structure present in typical plant cells and typical animal cells? Cell structure, plasmodesmata. Okay, so present in animal cells, they're not present in animal cells. So this will be wrong. Golgi body is present in both plant cells and animal cells, so this would be wrong. Centriole is not present and present. Actually, that's true. Plant cells do not have centrioles, so we would pick C in this case. Which row is correct for cellulose? Rotation of alternate monomers by 180 degrees. That is actually true for cellulose because of the way beta-glucose is shaped. So yes, that's true. Shape of molecule is branched. Actually, no, cellulose molecules are straight and unbranched. So I would say unbranched. And then hydrogen bonds between molecules, that is true. So that'll be D. Which statements about peptide bond formation are correct? The bond formation occurs between the carbon of one amino acid and nitrogen of the next amino acid after the amino acid detached from tRNA. Actually, no, they are going to form when they are attached to tRNA. So 3 tRNA molecules will be bound to the amino acid through 3 different sites. The first will be the P site, the second will be the amino acid site, and the third will be the E site. So the first 2 sites are going to be where the tRNA molecules are going to attach and they're going to have the amino acids, and where the amino acids are going to bond together. So 1 is true. 2, the bond formation occurs at the ribosome while the amino acids are still attached to tRNA and is a hydrolysis reaction. So the bond formation occurs at the ribosome while the amino acids are still attached to tRNA. 1 is wrong, as I mentioned, because as we mentioned, they are going to be attached to tRNA. But this is not a hydrolysis reaction, so 2 is also wrong. And the bond formation is important for growth of an organism when the bond forms, a water molecule is removed. That is true. Obviously, the formation of a peptide bond is important because we need proteins, but when it's formed, that's going to be a condensation reaction, so water will be removed, so it's D. The diagram shows a naturally occurring D-glucose and a form of glucose that can be synthesized in a laboratory known as L-glucose. So these two look like they're isomers. I mean, obviously they're isomers because they're both glucose with different structure. The enzyme glucose oxidase catalyzes the oxidation of D-glucose, and the enzyme cannot catalyze the oxidation of L-glucose. Why cannot not do that? Well, in this case, L-glucose does not fit into the active site of glucose oxidase exactly. L-glucose has a different structure to D-glucose, and so since they have different structures, they're not the same substrates, and the substrate is not going to be able to form the lock and key mechanism with the enzyme because, again, it has different shapes, so it will not be able to fit into the active site. So I would pick A. L-glucose has a different structural formula to D-glucose. They have the same formula, they just have different structures. L-glucose is a synthetic sugar. That's not the case. I mean, it's not even the reason why it can't fit into the enzyme. And L-glucose is a mirror image of D-glucose. Again, not a reason. 9. Tests for biological molecules were carried out on 3 solutions. Each solution contained only one type of biological molecule. The observations were as follows. Solution 1, test Benedict's test. Observation was blue to orange. So this is positive for reducing sugars. 2, Benedict's test after hydrolysis. Observation blue to red. So this is obviously positive for non-reducing sugars like sucrose. 3, Biarritz test, blue to purple. And so this is positive for proteins. And in this case, he's asking about sucrose, as I mentioned in amylase. Okay, which one will contain sucrose or amylase? Obviously, this one will contain sucrose. This one will contain amylase because amylase is an enzyme. And enzymes are proteins. Proteins will give positive Biarritz test. So 2 and 3 only. C. The effect on Vmax and Km when a competitive reversible inhibitor is added to an enzyme catalyzed reaction. Okay, so if you have a competitive enzyme inhibitor, the effects are going to be different to a non-competitive one. So let's just see and review quickly how the competitive enzyme inhibitor is going to affect the enzyme. So usually we're going to have something like this, a graph that shows the rate of reaction on the y-axis against the substrate concentration. As you increase substrate concentration, it will act as a limiting factor for until we have enough substrates that it's no longer a limiting factor and something else could act as a limiting factor. And at this case, at this point, we'd reach the maximum rate of reaction called the Vmax. Now, the Km value is supposed to be the substrate concentration at which we reach Km is the substrate concentration at which we reach half the maximum rate of reaction. So what's it called? When you add a competitive inhibitor, what happens is something like this. The graph looks something like this. It's going to eventually reach the Vmax, but it's going to look slightly different. As you can see, the enzyme is going to have lower affinity to the substrate as you keep on adding a great amount of substrate, but it's still a limiting factor. We need more substrate in order to reach that maximum rate of reaction. We need more and more substrate because the enzyme is just not able to see it anymore because the competitive inhibitor is going to come and is going to bind. It has a similar shape to the active site of the enzyme. It's complementary to the active site of the enzyme. And so what's it called? It's going to block the enzyme from seeing the substrate, and therefore it's going to take much more substrate to overcome that effect. So you could see how the affinity decreases, and so obviously in this case the Km will increase, because as you can see here, if we label this right here, the Km, as you mentioned, was this value at which half the Vmax would be reached. This is half the Vmax. Half the Vmax would be reached at a higher Km in this case. So this was initial Km, this is Km final, so it is increased. We still reach the Vmax, so the Vmax does not change, but the Km will increase. And you have to remember with me that affinity, once it decreases, that means Km increased. And in the case of a competitive enzyme inhibitor, nothing happens to the Vmax. So no change and increase. 11. The graph shows the effect of substrate concentration on the rates of reaction of 3 different enzymes X, Y, and Z. Now here we have what's the correct order of affinity of these enzymes for the substrates, starting with the highest affinity to the lowest affinity. As we mentioned, enzyme affinity depends on the Km value. So the higher the Km value, the higher substrate concentration at which we reach half the Vmax. So we're going to have a lower affinity in that case. So what's it called? As you can see here, the lowest Km, is going to, as I can see, it's going to be for enzyme X. So this is, let's say, half. This is 1500. This will be the Vmax. What's half of 1500? It could be something like maybe 750. I'm not really sure. I think it's 750. So that's something like right here. So now if we look at the Km value, it's really tiny. I think it's the smallest for any. Maybe it's like 100. Okay, so this means that it has a really high affinity. So let's see, this has the highest affinity, the substrate. Let's see this one. It reaches the Vmax at, let's say, 2000 and something. So let's go to 1000. This should be where we have half the Vmax, and at half the Vmax we should have the Km value at 600, which is quite high, and I think it's even higher than for enzyme Z. Enzyme Z, it has a low Vmax, but it has also a low Km value, a lower Km value than enzyme Y, and so that means it has a higher affinity to its substrate. So it will be enzyme X, then Z, then Y. X, Z, and Y, that's P. Okay. Which row correctly identifies the weak and strong bonds in tertiary quaternary structures of a typical protein? Okay. In tertiary and quaternary structures, so what we have here, typical type of bond. Okay. What's it called? Type of bond. Obviously, the strongest bond out of all of these would be disulfide bonds, and then what's it called? Hydrogen bonds would be quite weak in comparison. What's it called? What we have here is what's it called? only one option, sorry, two options which say disulfide bonds are strong. So obviously we'd pick this one because hydrogen bonds are never considered strong bonds. Okay, which row correctly describes hemoglobin? Four peptide chains, each containing a heme group. Iron ions can associate with oxygen, forming oxyhemoglobin. In each chain, hydrophobic R groups of amino acids point towards the center of the molecule at 50% saturation. Two oxygen molecules are transported by the oxygen molecule. That's exactly true. All of these statements are right. Yes, we do have four peptide chains. We do have four heme groups associated, one associated with each chain, and the heme groups can attach to four oxygen molecules. Half of that will be two oxygen molecules. Which processes always take place without the involvement of energy from ATP? Okay, so we have active transport obviously requires lots of energy, and cytosis in general requires lots of energy. Facet diffusion, however, is a passive process, and passive processes do not require ATP. Okay, the diagram shows the entry of a molecule X into the cell. We have molecule X, and you can see how it's entering by diffusion from a region of higher concentration to lower concentration, and since it's through a channel protein, this will be facilitated diffusion. Now, which row correctly shows the properties of molecule X and the effect of the concentration of ATP in the cytoplasm on the rate of entry of the molecule X? Okay. As you mentioned, this is diffusion or facilitated diffusion, so ATP should have no effect whatsoever because it's a passive process, and so we do not require ATP for it. So it does not have any effect truly. And then the type of molecule that we have here, since it cannot pass through the membrane, then this means it's a polar molecule, because again, the membrane here has a hydrophobic inner core, and therefore polar molecules could not really pass through that. So polar and has no effect on the entry, D. The electron micrograph shows some human blood cells. Which row correctly shows the net movement of water by osmosis and water potential of the cytoplasm X compared with the solution of the surrounding cells? Okay, so we have X. So you could see here again, shows the net movement of water by osmosis and the water potential of cytoplasm X compared to the solution of the surrounding cells. Let's see. Net movement of water by osmosis. I mean, you could see how the red cells look slightly, tired. They don't look really like like their biconcave selves. So I would say that the water will be moving out of the cell, and the water potential of cytoplasm of cell X would be specifically obviously lower than the solution. Higher than the solution, because that's the only way that the water will move out of the cell. So I would say C. Okay. A red indicator solution was mixed with agar, and the resulting solid was cut into cylindrical blocks. The blocks were placed in acid that turns the indicator yellow, and all other variables were kept constant, and the dimensions of the block are shown. So then he says we are given the dimensions of the block, and he's asking us to calculate the surface area to volume ratio given the formulas, and which row shows the correct surface area to volume ratio for each block and the time taken for the block to turn yellow. Okay, now we're going to do part of the calculations, and then the rest we're just going to do by solid understanding, because I don't have my calculator on me, and frankly, you don't really need to use the calculator for these questions as much as you think you do. Let's go ahead and try to solve this one. 2 pi r h plus 2 pi r squared. Let's start with finding the surface area of the cylinder of the block 1 and a volume of block 1 as well. Okay, so the surface area of block 1, we have the radius. No, we don't have the radius, we have diameter. To get the radius, you just divide diameter by 2, so this should be 3, divide this by 2, divide this by 2, so this will be 6 and this would be 8. Now if we use the formula, we get 2 pi r, so 2 times pi times radius of 3 times height of 3 plus 2 pi times 3 squared. So the surface area in this case would be, this is 9 times 2, which would be 18 pi, plus again 9 times 2, which is 18 pi, 18 plus 18 at 26 pi. Then to get the volume, you're going to get pi r squared h, so pi times radius squared times the height 3, which will be 27 pi. So we have 9 times 2, that's 18, 9 times 2, that's 18. So if we divide now the surface area by the volume, 26 divided by 27, you will get this 26 pi. If you divide 26 by 27, pi's are going to cancel, and you get approximately a 1-to-1 ratio. Now, moving on to the other blocks, as you can see, the blocks keep getting bigger. This means the surface area to volume ratio will just get smaller, and that overall, the rate of diffusion, or the time taken for the blocks to turn yellow, is going to become even bigger. So based on this information, let's try to figure out the answer without having to calculate all the other stuff. So we have surface area to volume ratio 0.75 to 1 or 1.33 to 1. The time taken should definitely be 4, as 4 is less than the other numbers on the other side. For block 2, yes, it definitely should be 5. and for block 3 it should be 11. Now let's look at the surface area to volume ratios of blocks 2 and 3 compared to block 1. Now block 2 cannot have a bigger surface area to volume ratio than block 1, so we can't have this as an option. And same thing for what's called block 3. It can't have a 2-to-1 ratio. That's huge. That's really great. So obviously it wouldn't have such a great time to diffuse, or like a great time to turn yellow. It should be a very small time if the surface area to volume ratio is this big. So obviously not. So I'm going to pick in this case C. As you can see, the ratio here is 1 approximately, 4 as the time, and it's less time compared to blocks 2 and 3, and a greater surface area to volume ratio compared to blocks 2 and 3, and also block 2 has a greater surface area to volume ratio than block 3 because it's also smaller than block 3. Okay, so the answer should be C. Moving on. Which metabolic process will be very active in a cell that has just completed cytokinesis? So it's now completely divided into 2 different cells in the case of mitosis. And in this case, it's going to now start with the G1 phase. So you have to remember how the cell cycle works. So the cell cycle we're going to have, so we're going to have a bunch of stages. G1, S phase, and G2. And lastly, we have mitosis. At the end of mitosis, we have what's known as cytokinesis, where obviously the cells are going to split. And in the G1 phase, what's going to happen is going to be the first growth period. So after mitosis, we're going to have small, tiny cells, and they're going to try to grow, and they're going to have, in case, like, let's say that they go under mitosis, then go under mitosis again, then they're going to need lots of things. They're going to need proteins, they're going to need, lots of enzymes to be produced, they're going to need organelles to be produced and everything. So they're going to undergo a bunch of things. One of them would not be DNA replication, because DNA replication happens specifically in the S phase. So directly after, I would say protein synthesis and ATP formation, so 1 and 3 only. Okay, the diagram shows a typical mitotic cell cycle and the point in the cell cycle that has been reached by each of 4 cells V, W, X, and Y. So you can see here we have G2, G1, S phase, and then mitosis and cytokinesis. Okay, which row correctly identifies the cells that match the 2 descriptions? DNA replication is complete, but the cell has not yet reached its maximum size. Let's see. This is for which cell? So right now we have W. W, you could see, is just finished with DNA replication because it's just finished with the S phase, and it's entering now its G2 phase, where it's going to check up on its DNA and make sure to synthesize more proteins and more energy storage molecules. Now for V, we've just finished the G1 phase. We've just finished production of, let's say, organelles and a bunch of proteins and a bunch of enzymes. And now we're moving on to S phase, where we're going to produce histones and DNA replication is going to happen. Now for cells at Y, they've just finished, let's say, telophase and are entering cytokinesis, where they're going to now split. These cells are going to split. And X here is done with G2 phase. The chromosomes are now officially condensing, and we're starting with prophase. So let's see what's happening. DNA replication is complete, but the cell has not yet reached its maximum size. That will be W. Preparation for microtubule formation is nearly complete, but chromosomes have not yet condensed. I would say that's X. Chromosomes have not yet condensed. Maybe they're starting to, but they would obviously have started formation of microtubules. So I would say V and W and X. Okay. Moving on, we have the graph shows the mean length of the spindle fibers during mitosis. Which region of the graph shows when all centromeres have detached from the spindle fibers? Okay, mean length when all the centromeres have detached from the spindle fibers. Well, in that case, I would say the length of the spindle is going to be quite low when the centromeres will detach from them. So I would say that the length would be at this point D. Okay, because let's remember together what's going to happen exactly. So during anaphase, the chromosomes, which are now like this, because they've split apart during anaphase, right? So they're going to be getting closer and closer to the poles of the cell, and so the microtubules keep shortening and shortening until there's no more of these microtubules, and so at that point centromeres are going to detach from the microtubules. So the length should be at its lowest. mRNA codons ACU, ACC, ACA, and ACG are all coding for the same amino acid threonine. Which anticodons could specify an amino acid other than threonine? Okay, let's see. Now this is anticodon, so it's supposed to be complementary to the codon. So let's find out all the all the anticodons for threonine, and then see which one is going to be the outlier. So we have ACU, that's going to be UGA. ACC will be UGG, ACA will be GAU again, and then ACG will be UGC. Okay. UGA is threonine, ACC is not threonine, not C, that's UGG, and UGC, we know this. So 2 will be, and what's it called? Wait, let's see. UGU, we've already done this, and then there's also UGC. Oh, UCA is not UGA. Okay, so yeah, 1 and 2 is my answer, B. Okay. Which bond formation does DNA polymerase catalyze? DNA polymerase catalyzes the formation of phosphodiester bonds between the nucleotides during DNA replication. So phosphodiester bonds between nucleotides. Let's see the other options. We have hydrogen bonds between nucleotides. That does not happen. Hydrogen bonds between bases actually happen quite naturally between hydrogen atom of one of the bases and an oxygen atom of another base. So no, this doesn't happen. D is my answer. In eukaryotes, the RNA molecules formed during transcription are modified by the removal of non-coding sequences. This is known as RNA splicing. It happens right after transcription. This is followed by the joining together of coding sequences to form mRNA. Okay, fantastic. So what happens basically, what are these coding sequences called? Well, the parts that are cut out are called introns, and these are the non-coding sequences. The coding sequences are called exons. So this will be the answer. Which row correctly identifies sinks for sucrose and transported by mass flow in plants? Okay, sucrose will be produced in the leaf in the form of glucose, which is then transformed into sucrose. So I would not say that a leaf would contain it, but a growing leaf would sure do need sucrose, because again, growing leaves still do not photosynthesize as much, do not produce as much sugar as they need, and so yes, they will be a sink. The root for will also be definitely a sink, and a growing shoot tip will also be a sink. The diagram shows the transverse section through a transport tissue in a plant, which row correctly identifies cell 1 and cell 2. Okay, cell 1 and cell 2. So the cell 1 is going to definitely be the companion cell, and cell 2 is going to be the sieve tube element, and this will be specifically at the point where it's ending in this point. You could see how, it's going to be, at the end where it's like a perforated ending. So cell 1 will be companion cell, and cell 2 will be the phloem sieve tube element. So this should be A. Okay. Which statements correctly describe the movement of solutes in the symplast pathway? Cell surfaces membranes regulate the selective absorption of solutes into the symplast pathway. That's kind of true, because let's remember the symplast pathway is the living part of the cell. Is the living part of the organism that the molecules are going to be transported through. So some things like the cell membrane, the cell cytoplasm, anything of this sort, not let's say intercellular spaces or the xylem or the cell wall, these are not really living, and so in that case that would be called the apoplast pathway. But if we're moving with the symplast pathway, then yes, cell surface membranes will regulate the absorption of solutes in the symplast pathway. Let's see the other options though. Plasmodes matter control the movement of solutes from the symplast to apoplast pathway. That's not how it works. The symplast pathway transports dissolved mineral ions from the soil that cannot be transported by the apoplast pathway. The symplast pathway transports, obviously mineral ions are going to be transported through xylem, and they're not going to be absorbed directly into the symplast pathway, so I would not say this is a correct option. And then the movement of solutes through plasmodes matter in the symplast pathway is prevented by the endodermis in suberin. Actually, the endodermis is going to have suberin, and so this forces the solutes to move through the symplast pathway. So I would say this is wrong, and so which statements help to explain why water molecules are forced to move through xylem vessel elements as a consequence of transpiration? Okay, water molecules form hydrogen bonds with cellulose in the walls of the xylem vessel elements in a process known as adhesion. Now, the formation of hydrogen bonds with the walls because of adhesion is really not the reason that water molecules are going to move through the xylem vessels. I mean, adhesion is something that happens as the water molecules are going to move because of transpiration. So I would say no. But the second one is water molecules form hydrogen bonds with neighboring water molecules in a process known as cohesion. I mean, you could argue the same thing, but actually cohesion is basically water is going to be moving together as one unit and mass flow is going to also be happening. So maybe this could be an option. So let's keep this. Let's look at the other options. Water molecules form ionic bonds with dissolved mineral ions, which help to keep the water molecules together in a continuous column. This is not the reason that water moves in a continuous column, really. Ionic bonds, they do water molecules could not possibly form ionic bonds with dissolved mineral ions. So I would say no. And again, it may form some bonds with them, But I wouldn't say that this is the main reason that they're moving as a continuous column. The reason they're moving as a continuous column is because of the hydrostatic pressure and the hydrostatic pressure gradient and the high water potential, also water potential gradient. And so what's it called? Maybe we could choose option B for this, but definitely not option C. Water has a high latent heat of vaporization, and this prevents the evaporation of water and design of vessel elements. What even? I mean, okay, yes, water does evaporate in the leaves during transpiration, but for it to evaporate in the design of vessel elements is not even an option. The temperature there would not really be high enough for water molecules which are moving together, which there's a bunch of water molecules in this case. It's not just one that's going to evaporate. No, there's a bunch of them, so it's really hard to evaporate inside anyway. So I would not pick this at all. I would say B is my answer. It means most correct out of all the options, even though it seems like a vague answer. The diagram shows the internal structure of a mammalian heart. Which letter identifies the location of the atrioventricular node? Okay, the atrioventricular node would be in the wall of the left ventricle, just above the just in the top, above the in the septum. We can say it's in the septum and everything. Okay, and then the diagram shows the pressure changes in the left side of the heart during the cardiac cycle. He says what happens at X. So at X is the start of ventricular systole. As you can see, the pressure suddenly increases, and so yeah, I would say definitely that ventricular systole is going to happen, and that's going to be when atrioventricular valves are going to close and semilunar valves are going to open. So atrioventricular valves close. Okay, so this is going to definitely happen here. Now he says here, semilunar valves open, semilunar valves open. So which do you choose? Do you choose semilunar valves or do you choose atrioventricular valves? Well, I'm definitely going to choose atrioventricular valves because just at this moment, pressure is going to increase. So just at this moment, we need to immediately shut down the atrioventricular valves, and then once we reach that peak, we need the atrioventricular valves to open. So I'm sorry, semilunar valves to open. So yes, I would say atrioventricular valves close. Which components of blood are present in tissue fluid? Phagocytes, some proteins, and sodium ions. Okay, let's remember what's in tissue fluid. So tissue fluid is approximately the same as blood plasma, except it does not contain any of the plasma proteins. So yeah, they have some proteins, not really big proteins though. Sodium ions definitely, and white blood cells definitely. So phagocytes would be included. In the lungs, movement of dissolved carbon dioxide out of capillaries occurs in one of two ways: by diffusion through endothelial cells of the capillaries, or by leakage through pores in endothelial cells of capillaries. What's the minimum number of times that the carbon dioxide molecule has been transported to the lungs? An red blood cell must occur across the surface membrane to reach an airspace in the alveolus. Okay, minimum number is going to be when there's a leakage. Now, how many surface membranes do you need to cross in order to to the lungs in a red blood cell? Must cross in order to reach the airspace. So we start with the red blood cell, right? So there's one red blood cell. Then we move on to the walls of the capillaries. Then we move on to the endothelial cells in the case where leakage through pores in the endothelial cells of the capillaries. So in this case, we may have, as we mentioned, pores in endothelial cells of the capillaries. Okay, and then we have the alveolus. Okay, so what we have here is, cell surface membrane number 1. Let's say we don't have to cross this, but we do have to cross the first and then the second membrane of, the epithelial cells of the alveolus. So we have 1, 2 over here. So we have to cross overall 3 cell membranes. What maintains the steep concentration gradient needed for successful gas exchange in the lungs? Airflow in the alveoli is in the opposite direction to blood flow in the capillaries. Airflow in the alveoli is opposite to the direction of blood flow in capillaries. I'm not really sure how this is going to maintain a steep concentration gradient. However, blood arrives in the lungs with a lower oxygen concentration and a higher carbon dioxide concentration in the air in the alveoli. That is true. And this may definitely help in the case of transport, because once we have a different concentration gradient, that allows for diffusion to happen, and this could lead to further steepness of the concentration gradient. Anyway, blood is constantly flowing through and out of the lungs, bringing a fresh supply of red blood cells. I think this is true. So I'd say 2 and 3 only D. Where is cartilage tissue always found in a human gas exchange system? Okay, cartilage is going to be found in trachea and the bronchi, definitely not in the bronchioles and definitely not in the alveoli. So yeah, trachea and bronchia. So D. Scientists compared the density of goblet cells in the lungs and density of mucus in the lungs of the three groups of people. So people who don't smoke and have lung disease, people who smoke tobacco but do not have the lung disease, people who smoke tobacco and have lung disease. The results are shown in the table. So what's it called? The goblet cell density, and you can see how this specifically increases with people who don't have what's it called? Lung disease. However, they do smoke tobacco, I would say, because they are smokers. Anyway, mucus density is going to be 26, and then smokers with lung disease have 37 and 15. So why would this happen? I think the case would be maybe someone who has lung disease, they would maybe have an autoimmune disease in the case where like immune cells are going to start destroying the air passage, and so some of the goblet cells are going to start being destroyed, and that's why they have less goblet cells compared to smokers who do not have lung disease. Okay, what is indicated by this data? There's a positive correlation or relationship between density of goblet cells and density of mucus. There definitely is, as you can see. Once this increases, like here for example, there will be an increase, okay, and so on. So yeah, I would say there's a positive correlation. Lung disease results in an increase in goblet density. Actually, no, it does not. It decreases it. Okay, there is an association between tobacco smoking and an increase in mucus density. That is definitely true. You can see how non-smokers are going to have a few less mucus density than the others. So yeah, I would say 1 and 3 only C. Which disease does Mycobacterium bovis cause? Cholera, HIV/AIDS, malaria, tuberculosis? I would say definitely tuberculosis. Mycobacterium bovis or Mycobacterium tuberculosis cause tuberculosis. Cholera, I would say Vibrio cholerae. For HIV/AIDS, it will not be a bacterium, it's in fact a virus. And then we have for malaria, it's going to be a protist known as Plasmodium. 36, an antibiotic inhibits the formation of crosslinks between molecules that form cell walls in the bacteria. Which statements explain why bacteria are killed by the antibiotics? The bacterial cell is destroyed by osmotic lysis. Okay, so bacteria are going to be killed because they're not cellulose molecules, so we definitely can remove this because again the bacterial cell wall is made of peptidoglycan. But then here we have the cell wall is no longer partially permeable. The cell wall was never partially permeable, so it also cancels out 3. Number 1 says the bacterial cell is destroyed by osmotic lysis. That is true. Usually lysis will not happen in cells that do have cell walls, but the case will be true once our cell wall is like damaged in the case of the bacteria over here. So yes, 1 only. Okay, moving on, we have scientists investigated the effect of increasing concentrations of the antibiotic on development of antibiotic resistance in bacteria. The scientists grew 4 groups of bacteria and added different concentrations of antibiotic to each group. The number of resistant bacteria and total population of bacteria were measured over the intervals of 24 hours for each group, and the graph shows the results. Antibiotic concentration is 0, antibiotic concentration is 90, and so on and so forth. So let's see. They keep increasing the concentration. You could see the population of bacteria in the beginning was actually quite high, but then what's the dotted line resembling, and so this is resembling the resistant population, this is resembling the total population. So you could see only a small percentage of the total population is resistant, which is the dotted line, and then the total population is quite high in general. That's going to be when it's at 0. But then once you increase the concentration just a little bit, you decrease the population of bacteria, increase the percentage that is going to be resistant, and then increase it even more, decrease the population further, and increase the percentage that's resistant, and then increase it further. You're going to see that most of the population here is going to be resistant, and it's actually going to be a very low population in the end. So which statements are correct conclusions that can be made from the results of this investigation? Increasing the concentration of antibiotic decreases the population of non-resistant bacteria at the end of 24 hours. At the end of 24 hours, let's see. No, actually at the end, they do end up increasing most of the cases. So I would say that it's not really true. Non-resistant bacteria. Sorry about this. Okay, non-resistant bacteria, let's look at the other ones. So at the end of 24 hours, we start here, for example, with 8. We end with approximately 7. We start here with 8, we end with approximately 4, we start with 8 and we end. That is actually quite true, I would say, because this is not going to be even included or considered in this case. So yeah, I would say that this is true. Let's focus here. The proportion of antibiotic-resistant bacteria increase with increasing concentrations of antibiotics. The proportion of antibiotic-resistant bacteria does increase with increasing the concentration of antibiotic. And notice here he mentioned proportion because overall it's approximately the same percentage. The same population, but the percentage of total bacteria is going to definitely be higher in this case. Increasing the concentration of antibiotic always increases the number of resistant bacteria. Not necessarily always. I mean, what's it called? Increasing the concentration of antibiotic here, for example. Let's see all the options. The increasing of concentration of bacteria always increases the number of resistant bacteria. I mean, you could see how all of the population is always at 2 and doesn't really increase much. It's just that the proportion of the total population has been it's almost entirely resistant now. So no, I would not really pick 3 as my answer. I would say 1 and 2 only. What is the correct sequence of events of primary immune response: T lymphocytes activation, B lymphocyte selection, plasma cell release? Okay, so let's remember how it works. So the antigen presentation by macrophages is going to be the first step. And then what's it called? Cytokines will be released by T helper cells, which are going to activate B lymphocytes, which will be differentiating exactly what's going to happen. So firstly, we're going to have macrophages which are going to, let's say, engulf or undergo phagocytosis. And once they do this, they are going to present the antigen that they just the antigen of the prokaryote they just killed. So in this case, they are going to undergo antigen presentation. This is going to result in the activation of T helper cells, which are going to release cytokines. The cytokines here are going to activate cytotoxic T cells as well as B cells, and then plasma cells will be formed and so on and so forth. So B will be my answer. Which statements about properties of antigen binding sites in different antibody molecules is correct? They are located on the light chains only. Are they located on the light chains only? No, because as we mentioned, the antibodies look something like this: 2 heavy chains, 2 light chains, and the antigen binding site will be right here. It's formed of both the light and the heavy chains. So not be true. They have hinge regions to give flexibility for their antigens. For different antigens, I'm not really sure that is the case. They have binding sites for receptors on phagocytes. They do have binding sites. This is the binding site, in fact, and it's not really found on the antigen binding site. And so I would say they're different binding sites. I would not really choose this. They have variable amino acid sequences for different antigens. That's true. Different antibody molecules have different, binding sites. They have differently shaped binding sites, and that depends on the primary structure and therefore depends on the amino acid sequence. So D. The diagram shows a stage in monoclonal antibody production. So you have the cancer cells, which are going to fuse with what? Well, obviously they're going to fuse with B lymphocytes. So yes, we're going to have B lymphocytes, which are going to fuse with myeloma cells, and these are going to produce the hybridoma cells. So this is it for this variant. I hope this video was useful for you. Thank you guys for watching. Make sure to check out, as I mentioned, the notes I have available for you. And if this video was useful for you, make sure to like and subscribe, and I'll see you guys in my next one.